And I was discussing if there is no nucleus in the RBC, then how it's going to stay fit in the circulations. So what are the internal structure, what is it made up of, and if you'll see a lot of variety of organisms exist in the system. For example, bacteria are there, fungi is there, plants are there, animals are there. So you will see there's a lots of compositional variation in their membrane, right? So if there is a lot of variation, because fish is living in the water, right? Amphibians both on the lands and water. So how they are coping with the environment? So is there nature gift any kind of things, extra lipid, extra proteins? And last time I was watching the question paper. They asked a very unique question, very simple question, but I think very few people were able to answer. Am I right? Have you seen this that question paper?
Actually,
I have no time to.
Yeah, I'm not so much familiar with this lesson.
I know. So don't ask me how I know, because I can guess. I was also a researcher 20 years back. So basic idea was to focus on the research at that time. Idea was to run the race. But then I realized after completion of my research that fellowship is very, very important. And if you are on the fellowship, you have a lot of scope, large number of scopes to get a good laboratories. You recognize this cell? This is a mitochondria. Mitochondria that has been isolated from the mammalian cells and stained with the green and fluorescent dye appear warm-like, right? And nucleus is going to be stained blue. Mitochondria are power generator that oxidize food. Already you are knowing this fact. Okay, so mitochondria, you have to know their structure. Then you can see there is an endosymbiont theory which suggests that
एक्चुअल में माइटोकॉन्ड्रिया कैसे दिख रहा है?
हाँ देखो ना, this is the electron micrograph structure, right? जो हम देखते हैं 3-dimensional and like this. So now how the bacterium internalized by the eukaryotic cells and become the part of eukaryotic cells, and they have not changed their characteristic features with the bacterium. They have a lot of similarity with the bacterium. So so this concept was given that eukaryotic cells may have originated as a predators, right? And predators means that can tell me what do you mean by the predators?
No idea.
Why this sentence you can say that they are evolved as a predators? Because if you say a lot of similar with the bacterial cell, mitochondria is there, photosynthetic cells are there, right? So that's why this seems to be right. Do you agree with this? Eukaryotic cell may have originated as a predator. So what is the role of microtubules? Microtubules helping in the distribution of the chromosome, right? So this is the historical landmark in the determining actual cell structure. A lot of effort have been done by the scientists. हमारे यहाँ स्टूडेंट क्या करते हैं? Lot of effort in the politics. Students are not deep focused totally. Very few students are able to focus and study and understand the meaning. Students are doing hard work, reading a lot, but they are not knowing and connecting the facts. So you can start with the Robert Hooke cells, and then Leeuwenhoek report of protozoa, and report of bacteria for the first time, then Brown about the movement of the cell nucleus in the orchids, Sildenson cell theory, Colliker described mitochondria in the muscle cell. That was a great observation. Fleming described with great clarity about the chromosome behavior during mitosis in animal cells. And काजल के बारे में भी पिछले साल ही पूछा गया था. Kazel and other histologists developed a stunning method that reveals the structure of nerve cell and the organization of neural tissue. Golgi first see and describe the Golgi apparatus, Boveri chromosome hereditary, palette porter electron microscopy, and then cytoskeleton. Robertson cell membrane, can do protein structure and X-ray crystal structure of myoglobin, right, and hemoglobin then. And Kristen de Dubé about cell fractionation of separation of peroxisome, mitochondrial lysosome, Petron and collaborators make the first confocal microscope. Lazarides and Weber uses fluorescent antibody to stain the cytoskeleton, and then GFP. In last 2 years, a lot of questions have been asked in the CSIR over the green fluorescent protein staining and the behavior of protein in the living cells. So just a quick recap about the cell biology, because I'm just coming to the membrane structure. Before knowing the membrane structure, you can see this ancestral prokaryote then divided into archaea bacteria and anaerobic ancestral eukaryote. And then from bacteria, you can see this line is moving, right? So this line is moving, mitochondrial line, and this line, chloroplast line. So chloroplast become parts of plants, and mitochondria becomes the part of animals, and then bifurcated into the fungi also, right? So non-photosynthetic bacteria totally divide up chloroplast, and non-photosynthetic bacteria and archaea totally have a different kind of structure. So about the model organism, a lot of questions I was just watching in different examinations.
सर स्लाइड एक बार पिछला वाला फिर से दिखाइए.
कौन सा मॉडल ऑर्गेनिज्म? ये वाला
सर इससे पिछला वाला जो था. हाँ यस सर.
नोट भी करते चलो है ना. ट्राई टू मेक द नोट्स. मेरे एग्जॉस्टिव डिस्कशन होने वाला है ट्रांसपोर्ट के बारे में, मेम्ब्रेन के अंडरस्टैंडिंग के बारे में, डिफरेंट टाइप ऑफ.
सर इसको सर एरोबिक बैक्टीरिया ना सर, एरोबिक बैक्टीरिया माइटोकॉन्ड्रिया.
नहीं वो तो एरोबिक एंसेस्ट्रल यूकैरियोट्स का पार्ट हुआ ना. ये देखो ना, एंसेस्ट्रल प्रोकैरियोट्स, एनारोबिक एंसेस्ट्रल प्रोकैरियोट्स.
समझा दीजिए.
देखो मैं ये कह रहा हूँ कि एंसेस्ट्रल प्रोकैरियोट 3 पार्ट में डिवाइड हो गए, ठीक. आर्किया, बैक्टीरिया एंड देन एनारोबिक एंसेस्ट्रल यूकैरियोट. तो ये एनारोबिक एंसेस्ट्रल यूकैरियोट गए, बाद में ये हो गए एरोबिक, ठीक है ना. बाद में लेटर ऑन जब ऑक्सीजन अवेलेबल हुआ, देन माइटोकॉन्ड्रिया शिफ्ट हुआ. माइटोकॉन्ड्रिया के शिफ्ट होने से बहुत फर्क पड़ा इन द एनिमल्स एंड फंजाई एज वेल. बट क्लोरोप्लास्ट इवॉल्व फ्रॉम बैक्टीरियल, वेरी सिमिलर टू बैक्टीरियल ओरिजिन, एंड दे मर्ज विद द प्लांट सेल्स. तो व्हाट आई वांट टू कन्वे दैट कि यहां तक जब तक माइटोकॉन्ड्रिया की एंट्री नहीं हुई थी, दिस वाज टोटली एनएरोबिक. बट जब माइटोकॉन्ड्रिया की एंट्री हो गई तो दिस वाज एरोबिक, ठीक है. एनिमल एंड फंजाई, ठीक.
ओके सर.
बात समझ आ गई? कोई डिस्कशन की बात ही नहीं.
सर माइटोकॉन्ड्रिया का जो है नॉन फोटोसिंथेटिक एरोबिक बैक्टीरिया.
हाँ, माइटोकॉन्ड्रिया का जाना, दैट बिकम द कॉज ऑफ ऑक्सीजन इवॉल्विंग. सॉरी, प्लांट्स की वजह से ऑक्सीजन आया, एंड देन एनिमल स्टार्टेड यूटिलाइजिंग एंड ऑक्सीडाइजिंग लॉट्स ऑफ फूड in the aerobic condition. So if you are before any kind of study, you have to select a model organism, right? So what kind of model organism you have to select? What is the basis? And you know, a lot of organism is being tested before confirming them as a model organism. So for example, I can show you E. coli for the purpose of molecular biology study. Right? Why this organism E. coli have been selected for the molecular biologists? Because the first and most important criteria is their division, cell division, how they are dividing, how they are growing, how easily they are growing, right? And their DNA should be very, very small in size, right? So E. coli was not only the microscopic, very small rod-shaped bacterial cells, easily found. You can isolate anytime, right? Easily can be cultured in the nutrient broth, right? And then you can see genetic instructions are contained in single circular double-stranded molecule of DNA, approximately 4.6 million nucleotide pairs long, and it makes 4,300 different kind of proteins. That is the unique feature of the E. coli. So that's why sometime in the molecular term they are called as the workhorse of the molecular biology, right? Constantly they are working, and you can know each and every aspect, how they are replicating, how they are decoding the genetic transformation, right? And how they are doing everything, how they are transporting, right? So because of so many properties, now a lot of studies have been done to confirm that really they are a very good model organism. For example, for the eukaryotic study, that question was, remember that question, about the telomere that was asked. Do you remember, Lakshmi, that question? So many students know yeast as a
Yes.
prokaryote, but the yeast is actually model eukaryotic organism, right? So obviously, all the facts and figure is going to apply related to the eukaryotes. Okay. So you have to think how you are going to think. And Drosophila melanogaster, if you are talking, very important to geneticists and biologists, right? Because after many studies, it has been found that they are correlating with various mammalian and eukaryotic developmental, and easy to culture, easy to isolate, very small, all that property making them favorite of the scientists. And another very favorite organism, multicellular organism, and first multicellular organism that had been completely sequenced was transparent. The body you can see very transparent, you can see the eggs, you can see the sperm moving, and everything you can see without killing them. And that is the beauty of C. elegans, right? So this is small nematode worm lives in soil, development from monozygote cell to the 959 body cells of the adult. And most individuals are hermaphrodite. That's also a kind of beauty, producing both egg and sperm, right? So that was the fact and figure. Zebrafish. Zebrafish are very popular model for study of vertebrate developments, right? So very small, very similar to, they are like fishes, convenient for study of genetics. They are also having a transparent embryo, right? You can watch cell moving and changing their characters in the living organism. So a lot of beautiful features are there, and making them as a popular model. Now about the membrane structure, what is the unique about membrane structure? So membrane, what is the beauty of membrane structure? They are enclosing everything.